Hi microfans, this is Lisa Schimmold. Welcome to 1 Minute Microbiology Techniques. Today I'd like to talk to you about the oxidation-fermentation test. What we're looking at is specifically how does a bacterium metabolize glucose, and the choices are either it oxidizes it, it ferments it, or it may not be able to use the carbohydrate at all. We're using what's called OF basal media with glucose. Here's what it looks like prior to inoculation. It's a semi-solid media, so it's a little soft in that tube. And when you run this test, you would actually inoculate 2 tubes of OF basal media for each organism that we want to test. You would inoculate by using your needle to stab the organism into each of the 2 tubes. And once you've done that, you will add a layer of sterile mineral oil to one of the tubes. Now we want to do this aseptically, and I'm not using any organism in this demonstration, but I would remove the cap, and here is my sterile mineral oil. You can either pour it by hand or you can use a pipette to add it. I usually am lazy and just do a pour. I don't want to touch the mineral oil tube to the tube that I just inoculated. And I'm going to pour, maybe a 1/4 inch layer into that tube. Recap my mineral oil. I would have flamed my tube if there was organism in here, recapped it. And actually, I really have more oil in here than I need, but that's okay. So once they're inoculated, I will have 1 tube that doesn't contain oil. We refer to that one as the open tube, meaning open to air. And then the tube that I've put oil in, we refer to that one as the closed tube, and I mean closed to air, so we're going to have anaerobic conditions in this tube. After incubation, what we're going to do is look at what happened in both of the test tubes. And we're looking for a color change from green either to yellow or possibly blue. Now, here's how we interpret the results. Oxidation can only occur in the presence of oxygen. So if only the open tube turns yellow, then the organism oxidized the glucose. Fermentation can occur both in the presence and the absence of oxygen. So if both tubes turn yellow, then that means the organism fermented the carbohydrate, in this case glucose. So let's take a look at the results. I have 2 tubes inoculated with E. coli here, and you can see that both the open tube and the closed tube have turned yellow, so that means that E. coli fermented the glucose. In these 2 tubes, I've got Pseudomonas aeruginosa. The open tube turned yellow, the closed tube did not change color. That means Pseudomonas oxidized the glucose. Now, in these 2 tubes, I've got Alkaligenes faecalis, and I'm not sure if you can see it there, but I'll just go ahead and tell you the open tube has turned blue because Alkaligenes really isn't able to use carbohydrates at all. The closed tube hasn't changed color. The reason for the color change to blue is that Alkaligenes is, rather than metabolizing carbohydrates, it is breaking down protein that's in the media, and the byproducts of that reaction are alkaline, causing the pH indicator in here to change to blue. All right, you guys, thanks for watching, and more later.